Here I'm going to record a short video, give an example of how to prepare your annotations using Genomer. Specifically, I'm going to show how you can generate and prepare your annotations for submission to GenBank. So first of all, I need to add a Genomer view plugin. So Genomer plugin view, and I'm going to be using version 0.6 or higher. And so I add this to my gem file, and whenever I edit the plugins in my Genomer gem file, I run bundle update, and that will make sure I have the required gems available. So here you can see here's Genomer plugin view and Genomer. If these plugins weren't available, they would be installed for me from Ruby Gems. So this is a simple example scaffold. So here you can see the sequence. It's just a single config. So there's the FASTA file, and if I look at the scaffold here, you can see it's just a single config scaffold file. What I'm actually interested in is the annotations, and that's what I'm going to describe in more detail here. So I have a single annotation here, and you can see it's just a gene, starts at 1, ends at position 2 on the positive strand, and it has the ID 001. So this is a very basic annotation which I'm going to use to illustrate how you can update your annotations. So I'll be using the command Genomer view table, which will generate the GenBank table file, and you can see it here. It starts at 1, goes to 2, gene, locus tag, and the ID here matches the locus tag for the gene. Now if I want to generate the CDS entries, I add this command generate encoded features, and that will now add the CDS entry in there as well. And you have here they have the protein ID. Now, first of all, I might want to name this protein. So for example, name PLB. So I separate these entries with a semicolon here, and it's a key value separated by a equal sign. So if I run that command again, you can see now the gene has the PLB added here, and protein as the product field added here, and it's capitalized P because that's the conventions for gene versus product, gene and protein names. I might want to add, so then what if that's the name of the gene, I might want to add a product. So for example, the product is pilus assembly ATPase. And I run that command again, and you can see the function, the product field is mapped onto the function field here in the protein. So here I have pilus assembly ATPase added here in the CDS field. I might want to add a note, for example, involved in type for pillins. Run that again, and you can see this note field is added in here additionally. So you can see that's how I can populate the fields in my GFF file here, and they will be correspondingly added to my generated GenBank table file here. And you notice that, for example, name is capitalized, as is note, whereas product is not. And that's because in the GFF3 specification, they have a controlled vocabulary, and these are the capitalized words. So if you're using a capitalized words, that should be from the GFF3 vocabulary, and you can add anything you want here if it's lowercase. And so product is a field that I've added that Genomer will read from your GFF3 file. So I can add it. There's a description here in the man pages. So if you ever are unsure about something, I try to document everything as much as possible. So Genomer view, Genomer man, and then the name of the plugin I'm interested in. So Genomer man view, and you can see this is the man page. And if I go down, you can see here how there's a section describing GFF 9th column attributes. And so here's ID, name, note. And so these are the GFF defined attributes you can use. And then here you have Genomer attributes. So these are ones I've added myself, and so I'll cover these a little bit more. So dbxref is a database cross-reference, so you can if you wish, you can add cross-references to other databases. I'll add an extra annotation here to illustrate that, so this line doesn't become too long. So for example, dbxref, and this will be saying, say you have a gene ontology cross-reference, and then I have another one, say be dbxref equals interpro. So assuming I've added those correctly, they should now appear. Yeah, so this is the additional one I've added. And so here you can see both of these dbxref have been mapped. So GFF expects this to be in one format, but the GenBank table expects it to be in this format. And so Genomer can just be used to map between those two fields. So I should just make sure I change this here. You can add EC number is another field you can add. So I add EC number equals 1.1.1.1. If I run that, let's change the ID as well. Run that. Did something go wrong there? Yeah. For some reason it became. Here we are. So this is the extra one. I have the product here, alcohol dehydrogenase, and EC number here. And you'll notice that here I've added name in PLC, and here it becomes PLC becomes the product. And then here I've added the product, and it still maps to the product. So I try to disambiguate wherever possible. So if I add a name in here, for example, I'm not sure what the 3-letter code is for alcohol dehydrogenase, so I'll just put that in there. And then I run this again. So now the product, this field here becomes the product, and the description alcohol dehydrogenase gets shifted down to the function keyword. So so I try to disambiguate those fields intelligently as possible. Prefer name first, and then if product is available, shift that down into the function field. And there's one more thing that's important to talk about. If I have, say for example, for example, a 16S ribosomal RNA. Now if I have this in here and I run this command again, now this generates a CDS, and obviously a ribosomal RNA gene is not a protein coding gene, and so you can fix these in Genomer by adding feature type equals RNA, ribosomal RNA, and I run this here. And now you can see the correct feature type is added here. And it's worth describing where these are here. find these. Here you are. So I have a specific list of ones that are included in Genomer, because these are ones I know of at the moment. If you have any additional fields, so it's RNA, tmRNA, tRNA, miscRNA, if you had any other additional entry types you think should be added to Genomer, just let me know and I'll add those to Genomer. But that's how you add a non-protein coding gene and have it output the expected output in Genomer. So one final thing worth talking about is you might want to change the IDs here. So if I add a prefix, for example, I'll just choose 3 letters at random, IRB underscore. You can now see the IDs have been changed here. They've been prefixed with this prefix, so they've gone from being 004 to IRB underscore 004. When you submit something to GenBank, usually they provide you with this prefix, which you have to add to your annotation. So that's how you can do that in Genomer. If you want to, you can also reset locus numbering at a different value. So for example, I want my annotations to start at 60. I did that correctly. No, I spelled that wrong. And now you can see my annotations start at 60, then it's 61, and so forth. So that's how you can change the IDs if you want them to start at a specific number. And finally, you often have to prefix your CDS annotations with a specific unique ID that identifies your lab research group. So for example, I want to prefix this GNL bar, Genomer bar, and run that command. And you can see now the protein IDs have been updated, so they still have the corresponding gene ID, but they also have this unique prefix which you should add when you submit your annotations to GenBank. So I hope that makes it clear how you can define your GFF annotations in Genomer and then use that to create the table file for submitting your genome to GenBank. As always, the command is, if you want more information about this, Genomer man view for the view plugin, and that contains a lot of extra additional information about the commands you can use to specify your annotations. And if you have any questions, my website is next.gs, or my email address is mail@michaelbarton.me.uk. Thank you.